Yeah, my name, for those of you online, my name is Drew Harvell. I'm a professor emerita from Cornell University and a resident researcher here and an affiliate faculty at the University of Washington. And in our focus on the ecology of host pathogen interactions, one of the really useful ideas is to think of them as indicators of changes in ecological interaction. So one of the kinds of indicators of ecosystem function that Manu mentioned this morning. In addition, for some of the work we're interested in, we're thinking that the health of these organisms, because the signs show so visually, can also be good clues for finding climate refugia and managing these resources. So my work has involved hosts from corals. We've done a lot of work on coral reefs. More recently, we're working with seagrasses here on the West Coast and a whole cadre of sea star species on the West Coast as our hosts. We've worked with a range of pathogens, including protists and bacteria and fungi. And I think that box or that circle for the environment is obviously one of the most important here for what we're talking about, because we're dealing with a changing environment. And in particular, the kind of change that you're all studying, the heat waves, is something that is kind of a vital trigger of some of the outbreaks that we're studying. So we've been at this kind of a long time before we were even talking about heat waves. We've been identifying that warming trends in the environment have been triggering various kinds of outbreaks. For us, first it was corals for many years, and now more recently, some of our temperate organisms. So if anybody's interested in this realm of study, there's a whole bunch of papers over the years by a large group that we've been working with on this. What I want to talk about today is work that's sort of leading to our managing our coastlines better for more resilience. And so I'll talk about climate and health-driven declines of corals, sea stars, and seagrasses, and particularly the temperature links in those. Some of our work is quite synoptic in terms of being continental scale and having a very detailed data set. And I got pretty excited listening to some of Manu's approaches, thinking that we're really going to be able to delve much more deeply into some of the more mechanistic work behind the effects of some of these heat waves. And then a lot of this is going to be leading towards a quest for resilient refugia. After my lecture, if we have time, there's a whole bunch of things that it could be interesting to discuss. It's going to be kind of up to you, but certainly the forecasting and hindcasting value of the kinds of work that you're all going to be embarking on, how this can lead us to figuring out better protected areas. We're going to be moving into the realm of assisted evolution because we're exceeding the limits of some of our organisms in today's oceans and other ocean priorities. So we're not the first to figure out that there's and study temperature sensitive diseases of marine taxa. And I'm not going to go through all of these, but I do kind of want to point out the top three. These are all organisms that live in one way or another on the bottom in coastal environments. And virtually all of their diseases are very apparent. And that makes them good as sentinel or indicator organisms, because it was interesting, some of Manu's data this morning on fisheries populations, that's a really downstream effect or impact. Picking up the health of an organism before everything starts to die could be years before you get a population level impact. And so in terms of an early warning or taking a pulse of the resilience of a population, this could be a more immediate way to do it. So this was just a paper we had a few years ago led by Jeff Maynard, kind of summarizing all these marine taxa and their temperature sensitivities. And then his was also one of the early studies to try to actually do forecasting for disease events for something that's economically relevant. And so in addition to summarizing a bunch of marine disease outbreaks and their temperature sensitivity, he and the group, and Mark Eakin's one of the speakers here, he was part of our group then, went forward to try to figure out what the seasonal outlook would be for lobsters in Long Island Sound. And for lobster shell disease, there's a 12-degree kind of threshold. And once the temperatures go past that, they're more likely to develop this disease. And so they used that example, and that's what you can see. Can you see my cursor? Yeah, that's what you can see in that second plot there is just probability the bottom temperatures will be greater than 12 degrees C for greater than 7 days. And that was for a particular year and time. It was kind of a near-term forecast on that. So if you're interested in that, this was kind of trialing some of those early approaches to forecasting these effects. Another issue that we've dealt with kind of in the broader sense is trying to understand what baselines would be for marine disease. We're often asked, are we seeing big increases? And the answer now is pretty resoundingly yes. 10 years ago, it was a little less clear. And so when Allison Tracy was a PhD student, she worked in a meta-analysis to develop baselines for marine disease. So this was a pretty synoptic analysis of all marine taxa, from sea urchins to marine mammals to fish. And what she and her team did was actually to look at the records from 1970 to 2013 for all these organisms and what was the incidence of disease that had been occurring in the field. And so this gave us some kind of a baseline. And then they went on with the corals, which were at that point at about 2% of the reports at the time were from disease. They went on with the corals to show through the temperature anomaly data that the increases in disease were actually related with temperature. And so that was, again, the first time for a fairly long time series to be able to nail down a linkage between the environmental change and actually an increase in infectious disease. And I just included this slide as an example of one. Actually, I'm going to be at some meetings in Mexico in the fall, dealing still with this stony coral tissue loss disease. Some people call it Skittle D. And you can see this is a real killer. It's affecting a wide host range of corals. And that bottom plot shows you from February 1st to March 17th, so a little over a month, that this disease has expanded as the white spots of dead tissue and killed all the living tissue on the coral. And so this continues to be an enormous management issue in the Caribbean and is very temperature sensitive. And then I think probably everybody knows the kinds of impacts to corals that we've been seeing. And so if you don't, here's a more recent paper by Ove Hogoldberg in Science. And I mean, it is pretty impressive to look at this plot from 1910 of the average global ocean temperature anomalies from February to July each year, and to see how dramatically this has increased through time. So that's kind of a sobering effect. And then what's plotted here in these little histograms is kind of our standard way of looking at heat stress on coral reefs, which is, or temperature anomalies, which is degree heating weeks. And so what's plotted here is the degree heating weeks, number of days degree heating weeks exceed 4. And you can see again, since 1990, this has increased dramatically. And then 2023 was so hazardously lethal for corals you can see here that the sea surface temperatures just went off the scale that year. So that's all I want to say about corals, because our current work is not focused on coral reefs. And yet it may be, how many of you here are doing any projects around corals? Is any, yeah. Okay, great. So those are obviously really important projects. Again, it's a foundation species that's being heavily impacted. I want to mention some of the work we've done with the keystone species. This is the sunflower star, and I like to point it out because it's here. And I hope later in the course, we can arrange for you to get a little bit of a tour of the captive breeding facilities here for the sunflower star. This is a species that's been driven to endangerment just from infectious disease. And this is our paper, Disease Epidemic and a Marine Heat Wave are associated with continental scale collapse of this pivotal predator. And the reason it's gotten a lot of attention is because it's the largest marine wildlife epidemic that's ever been recorded. And you're going, no, that can't be right, right? There's got to be more important things, bigger things than that. Well, the reason it's so big is because over 20 species were affected. So it was a very wide host range. And the geographic range was from San Diego all the way up to Alaska. And we are still contending with seriously reduced populations of several of these species, in addition to the sunflower star, which is now on the endangered species list from this. And so I want to just point out a little bit more about it. Because this was, again, a case where there was a climate linkage. So we're currently working, we have with the Sunflower Star Recovery Plan. And I said the captive breeding is ongoing here. And we're also involved in figuring out what the causative agent is. It's caused by a microorganism, and we're in kind of final stages of figuring out what that is. But this has caused a 90% global decline in this species. And you're going, well, you know, I mean, species come and go, right? So no big deal. It turned out to be a big deal because this species is controlling sea urchins. And you're like, well, sea urchins. But sea urchins can control kelp. And so the removal of this top predator caused sea urchins throughout California, but also into Oregon, Washington, and British Columbia to explode and totally mow down seagrass meadows. And so now we're having a problem with the seagrass, the kelp meadows being completely destroyed by the urchins because of the removal of this top predator. And so that's why there's a big effort to try to get it back. I just want to show you a little bit about sort of the heat wave part of this. So this is some of the data that we have, and I was going to show you a video, but it's a little sketchy with the links that we have, so I'm not going to run that. But I'll put these lecture notes up, and if you want to later see it, or we can look at it later. What you can see here is these were the populations of this star starting in 2004. So this was citizen science diver data. And then when the epidemic began in 2013, 2014, the populations tanked. And this is Washington, Oregon, and California. So the British Columbia data aren't shown here. And you can see this was an extreme decline throughout this entire population. Our team was also able, and I'm not the one doing the statistical work on this, but our team was also able to link these declines with the warm blob. And you heard about that this morning. And so I reproduced one of Manu's my favorite of Manu's slides here, the 2014-2015 ocean heat wave, where he showed in, we have to get rid of me. Does that show there? Yeah, I'm kind of in the way. This shows 24, I think it's 2014 here, and then how it expanded through the entire coast. And then these plots, which are in the paper if you want to go look at it, basically show British Columbia, Washington, Oregon, and California. And then these are the sea surface temperature anomalies. And that's a fairly simple calculation of temperature anomaly compared to some of the things that Manu is talking about. And what you can see here is in 2015, here's the blob. So it's showing a very marked departure from any of the other years in British Columbia, in Washington, in Oregon, and in California. And so again, that just kind of gives you a sense of the on-the-ground temperature stress we get in these kinds of events. And this is also an anomaly I'm going to come back to in talking about the eelgrass. So this is our team that's currently working on figuring out the infectious agent. And I'm not going to talk about that anymore here because it's really not within the realm of the heat wave work. But we're hard at figuring out what the microorganism is involved. And I'm going to skip that part. We're also working on the immune system, trying to understand what are the components of immunity that's failing in the species that's endangered. Why are some of the others bouncing back and this one's not? And we think the clues are in the immune system. So this is a transcriptomic study of the immune system. And of course, heat is an important part of the study. Okay, so I wanted to show this. This is what we call the Pacific Heat Dome. And this was an event that occurred in 2021. And hopefully that's actually playing. And I'm kind of interested because I think in terms of the work you guys are doing, you would classify this as a terrestrial event, right? And yet this was incredibly destructive for our coastal regions. So if you look at the extent of that anomaly and its geographic scale and its coastal impacts, you can see that we also need your help looking at some of the ones that might start in the oceans and then move over and crash into land. This one was really quite destructive of our eelgrasses, and I'll come back to that in a minute. So we started studying the disease ecology of eelgrass in 2012 because we had just come off working with corals and watching them dying from heat-sensitive diseases. And here we were in Friday Harbor. We thought, well, good, we're past that. Things are going to be better. And then we discovered that there was a disease of our most important habitat-forming species, eelgrass, that was temperature sensitive. And so we said, we're going to start monitoring that because we really think there are going to be warming events in the next decade that are going to affect us. And unfortunately, we were very right. A decade of our research has shown major outbreaks of the disease and dramatic declines. And so that's what I want to tell you a little bit about. So if some of you end up for your response variable or your indicator system, if you're studying heat waves, studying diseases, there is a lot to unpack. So we work pretty much with some of our climatologists that actually, help us do the work. And then we're actually doing the diagnostics and the biology of the seagrass wasting disease. But I'm going to give you just a little picture of kind of what's under the hood of some of this. So this is nice. We have lesions, right, that you can see on the surface of the plants, and the plants are attached, so they're not running around. And so we can do spatially explicit work. Over the decade, we've worked on developing a lot of diagnostics. We can culture this particular agent. A lot of the ones that are affecting marine taxa, you can't culture. And it creates these very distinctive lesions with black edges. Now, the black is actually the plant response to the pathogen that's killed this tissue. The living tissue is green. And so this white is tissue that's already been killed. And this particular lesion is advancing on two fronts. And eventually that whole blade will disappear. It will just die. So this is the bug. This is a protist. It's alive. So you can see it in its culture dish. It lays down a mucus net, and then these little cells swim through the mucus net and eat away at the plant tissue. And this is what it looks like in histology. All of our undergrads can culture this easily. So we send them out, they collect a diseased plant, they can bring it back, and with the right culture media, usually they can get it in culture pretty readily. And that's an amazing, great thing. We can also do these kinds of experiments. So this is an inoculation experiment where Morgan and Phoebe inoculated the plants. So this is what it looks like on day 5, 12, and day 20. So we have complete confidence that this is the correct pathogen that causes those lesions. We also have qPCR tests that tell us how many cells are in every one of those lesions, so we can monitor the severity. I'm kind of showing you what's under the hood behind doing this kind of analysis. These are our field surveys, and this is 9 years. Now we have 10 years of eelgrass meadow density and disease. And so this shows you the wasting disease prevalence. We started in 2013. Some of our populations had almost no disease, whereas others actually were showing some significant levels. But irrespective of that, everything peaked after the warm blob. And these are the periods. Remember, we've talked about, Manu talked about it this morning. I mentioned it earlier, between 2014 and 2017, when we got hit with anomalously warm sea surface temperatures. And so all of our populations spiked up to almost 90% of the plants with disease. In turn, what happened is the population started to decline. So this is the density of our plants. And now you can see they're at very low levels after a decade of repeated outbreaks of this disease and other temperature stress on these populations. And these are all our islands nearby. So Beach Haven, False Bay, these are on Orcas, Shaw, and our island here. One thing, if you're thinking about like indicator organisms, like what are you going to use to sort of compare the impact of your heat waves against? You want to be sure your data are good. I'd like to tell you how good our data are. For this, we have between 3 and 6 20-meter transects at each site, and then multiple times. So there are thousands of plants underlying the data here and repeated surveys.
I think Beach Haven got hit harder a year before the other sites you haven't got hit.
You know, Beach Haven has been a real mystery to us because it was one of our most beautiful sites. It was very high density. You can see here that it had over, 250 shoots per meter squared, and it just collapsed. And it was preceded by this enormous outbreak that went on for years. You can see Beach Haven just got trashed. I'm going to show you the drone footage. Olivia just finished mapping with drones. There's no eelgrass on that entire beach. The entire intertidal meadow is now gone from that location. Similarly, pretty much the entire intertidal meadow is gone from False Bay, 4th of July. And there's a few plants remaining at Indian Cove and North Cove. Okay, so these have been complete catastrophic losses of all the intertidal plants. And I don't know why that one went first. I mean, it wasn't a particularly heat stress location. Yeah. It is closer to the Fraser River. If there was some kind of a stress coming from that. Yeah.
Notice that there's like some sort of tipping point for these zostera beds where it's like over this much coverage on the blade leads to catastrophic die-out?
That's a very good question. If you look at sort of Beach Haven again, so you see that starting in 2015, it basically took off. There were like 5 years of really high levels. 90% of the plants on all of our 6 transects were sick. So, you know, that's a pretty good indication that what was going to happen to follow it. Just catastrophic decline. Yeah. I don't know, Olivia, any thoughts on that? Like tipping points?
Question. I don't think we have a hard number necessarily, like 80% of the leaf is covered, then it's gonna die. I should know, and maybe Drew's gonna get to this too, there are different strains of the pathogen that vary in virulence too, so you have to kind of factor that into the equation. So if you infect plants from site A with a strain from site B, they do worse than if you infect with a native strain. So yeah, I'm not quite sure. That's a really, really good question, but there's a lot of different factors that go into the resilience of these meadows.
Yeah, so it would be really nice to look at to compare the intertidal and the subtidal ones and look at, you know, the marine heat waves as well as the atmospheric heat waves, right? Because the intertidal ones are getting impacted by both, and the subtidal ones may be getting impacted more by them. Okay, so obviously, by the way you're nodding your head, that's where you're going.
That's such a good idea, Chuck. Well, I'll show a little bit of that. Yeah, get to that. And then again, I love this slide of Manu's this morning, just showing again how that just incredibly ecologically destructive heat wave developed and then just sat on our coasts. It was, and you're going to hear more about that from Dr. Julia Parrish this afternoon with the seabirds too. Here's a little bit of the drone footage. So in our more recent study with the Smithsonian, and I'll show you the latitudinal scale. So the work all started here. We're the center of gravity for this eelgrass work here in San Juans, but we expanded to the north and south in studying this. But just before we leave the San Juans, I wanted to just show you the horrible impacts we've had. So this is what that seagrass meadow, and these are places you can go to. That's our beautiful beach haven, shown here in 2019. There's plenty of eelgrass. 2020 there was eelgrass, and then by 2021 it basically all fell apart to the point where there were no plants on any of our 6 transects, both the shallow and the deep intertidal transects. And then the same thing, 4th of July Beach, which is, I'm sure you're going to get there in the time you're here. Again, it used to be covered with eelgrass in 2019, and it's a wasteland now. There are no plants, hardly at all. And then just to really bring this home, this is this beautiful beach haven. Already by 2020, our shallow transects no longer had very many plants on them. You can see the edge of the bed is receded here. The deep water transects, the deep intertidal transect still looks great in 2020. It's all gone in 2021. I'm only showing you 2 of the transect, all 6 of them are gone, and multiple other sites show the same thing. So this is a really huge impact in a very important habitat for us. And so we went on to develop a machine language learning tool with our colleagues at Cornell. This was developed by Brendan Rapazzo and Carla Gomes in the Institute for Computational Sustainability. And this is, I'm getting a little back here. This is not just, okay. This is not just an image analysis thing. This is actually an AI learning, machine language learning tool that took years to train to be able to recognize this. But the reason we did it is because we wanted to run 900 images all at once, and we couldn't count on any other mode of diagnostics or subjective error in that. And so in order to be able to do this latitudinal scale study with nodes from San Diego all the way to Alaska, including our sites in the San Juan Islands, we needed to use that tool. This paper is the one that if you want to read or we might discuss that I've suggested. This was led by our then postdoc Lillian Aoki. But you can see Olivia's part of the team there. And there's a whole range of other colleagues at these different nodes. So what we're plotting here is we wanted to know, are we the only ones having a problem in the San Juan Islands? What about to the north where these populations are more pristine? What about to the south? This population is, this species is interesting because it has such a wide latitudinal range. You can't do this with very many taxa. And what you can see, we're plotting the cumulative positive temperature anomaly. So obviously the temperatures, are higher in San Diego than Alaska, but maybe the anomalies aren't, right? And so against the wasting disease prevalence, the percent of infected individuals. And in this year, this was 2019, these teal spots are most of the northern populations had a temperature anomaly in that year and had elevated disease outbreaks. The green down here was Oregon. They were kept cursing me because they couldn't find lesions. They were supposed to be out collecting plants with lesions. They couldn't find anything on any of their plants. And it turned out to be an anomalously cold year there with very little or no disease. And then the size of the lesion area, this is wasting disease lesion area, also was reflective of the level of the positive temperature anomaly in that year. And again, I want to emphasize for this kind of study, we had an enormous amount of data, 2,400 plants per time, 120 transects over this entire range. And we have four years of data, which is about to come out now for more years. Again, this shows that disease increases with warming events, not absolute warming, which I don't know. I mean, you can take that as a glass half full or empty. I'm sort of viewing that as a positive sign because it suggests to me that plants are adapted for the environment or temperature in which they live. That means we should be able to move southern plants further north. They're not able to adapt quickly enough because these heat waves are coming too fast and too intensively. But if we move in some more resistant populations, we may have some more chances. So we're kind of at the point of thinking about that now. And so again, this just shows that the wasting disease increases with the temperature anomaly. This is some of Olivia's data showing that the blade growth decreases precipitously with the disease severity, which probably wasn't a surprise to you. I'm not going to talk about this so much now. I thought after I go through this particular lecture, we have some time for questions and discussion. And I thought that would be the time to really talk about what some of this temperature data looks like. But these are basically the temperature plots: Alaska, British Columbia, Washington, Oregon, California, Bodega, and San Diego. Again, showing the absolute temperatures and the sea surface temperature anomalies. And I do kind of like looking at these. I mean, San Diego, you have an 18-degree temperature all summer long. Alaska, it's closer to 12 degrees. So, and then anomalies picking up through time. So, yeah. So I'm going to come back to this. These are graphs from that Aoki paper. It has a lot of good detail to discuss, including how to develop machine language learning, how to get good habitat organism data, comprehensive enough, the value of a wide latitudinal range. I'd be interested in your thoughts about the value of disease as a sentinel. I mean, that's something that we think is useful and how best to do that. But so I'm going to come back to this, but I just wanted to show it here. So, the data that we have is we're mostly using satellite for the anomalies. We have to use the satellite data because that's the only way to get the 20-year comparison to calculate a temperature anomaly. But the problem that we have is that we're dealing with intertidal populations. And so all of the temperature stress is not captured by the sea surface temperature data. And this is one of the issues that Olivia is going to be dealing with in her project here. And I just show a plot from Lillian's earlier paper that was in oceanography in 2023, where she plots the data on, sorry, that's July 21st. This is again that heat dome, that enormous temperature stress. We had temperatures in excess of 100 degrees on my deck here and also on the coast. So this is what you get from the satellite data, but this is from the logger data, the hourly in situ temperature data. So we're picking up enormous temperature spikes on the beach. And so one of the big challenges is how to integrate those two data sets. Yeah. Okay. So you're like, well, it's not too bad. You know, you got plants on the beach, you can mark them and follow them. Well, but remember, we're studying a host pathogen interaction. And so it is kind of relevant to know how it's transmitted and whether there's activities or conditions in the water column that might be affecting the way these diseases are acting. And so the sort of bad news on this one is it's highly transmissible in the water column. And this was a paper just published by one of our team members who just finished her PhD this week. Her whole dissertation's in now, Morgan Eisenlord. And what we did is we put out sentinel plants in the eelgrass beds, near the bed, and then several hundred meters away from the bed. And so these are plants that don't have any signs of disease when they're put out. And so after 3 weeks out, the levels of disease were the same across that range. And so that's indicative of very widespread transmission of this disease. And now, so we're having to develop eDNA methods to monitor the levels of the disease in the water to see if it's predictive of what kinds of levels we get on the beach. And that's, I don't know, maybe that's bad news again, because we're actually finding very high levels in the winter, which was not at all what we expected in the water. These are our average copies of the pathogen in the water in January, February, and then throughout the year. And what we're finding is spikes in the water in the winter may be setting up the disease situation in the spring and summer. And so there's a whole phenology here of when is the worst time for heat shock to happen in terms of activating a big outbreak. And so we're
the cycle of the pathogen.
we know a little bit, but they do it releases zoospores, but we don't know where these are coming from. We don't know what the source is of these at this time of year. This really was a surprise to us. It wasn't really Olivia's favorite thing to be going out in the dark in the winter to collect these samples, because that's when you have to collect them in the winters. The low tides are at night and the weather's nasty. I think I'm going to, well, I'm going to give you the whole story. You might as well know all the things we're dealing with. This is another project, what we found. This is again working across this entire range. And we wanted to know what were all the controls on the levels of disease. And so obviously temperature was one that we found early on. Through some of Olivia's work, we knew there was a possibility that the grazers could be spreading this disease. And she has a paper that's in Review in Ecology about this. And so I just want to show you that the grazers in this study over this entire range were associated with an almost 30% increase in disease. And it's mediated through temperature. So this is a structural equation modeling analysis. And you can see here June temperature anomaly and June mean temperature are important as indirect effects in the model. They're driving the epifauna, which is, that's what that big critter there is. We call that an epifauna, right? Driving their numbers, which then affect the lesion areas. Similarly, the June mean temperatures affect the epiphytes, which can have some effect on disease. So we're kind of working through the multiple levels. I thought it was really interesting to think about, Manu's struggles with atmospheric versus ocean different levels and how those relate. And then on top of that, we're dealing with multiple levels here as well in predicting these causes of increased disease. The bottom line is we've got big losses. And so this is where we need you to all be helping with the forecasting and the understanding of basically the life history of these heat wave events and how they're affecting our habitats. And so this gets back to Chuck's question. This is work that Olivia did. I don't know, do you want to describe it, Olivia? Do you want to?
Either way, yeah, I can or you can, Drew.
Go ahead.
So for this, this kind of gets to Chuck's earlier question about disease in intertidal meadows exposed at low tide versus those in subtidal.
You should turn up, you should unmute,
Oh.
and you should mute,
Oh, okay.
so that the people.
Maybe I'll just do it because if I've got to stop sharing to unmute.
Oh, okay.
I can.
Oh, okay.
Okay. Okay, hopefully folks online can hear too. I'm getting some feedback. So here we were looking at intertidal and subtidal eelgrass meadows spanning from Alaska down through South Puget Sound. And we expected that we would find lower disease in the deeper subtidal meadows compared to those exposed at low tide in the intertidal. And that's exactly what we found in 2 years of surveys across pretty much all of our geographic regions. And we also found close associations between ocean warming, the sea surface temperatures, and higher disease levels across those years as well. So, and I'll provide a little bit more detail on that tomorrow. But this really highlights this is a very tractable system for studying impacts of heat waves on disease outcomes for our local meadows and local and beyond, I should say. Yeah.
Okay. So again, just to point out in Olivia's study that she did a range of work in Puget Sound, the San Juan Islands, really pristine locations in British Columbia, which actually we had thought there wasn't going to be any disease. We thought, oh, we'll get there first before anything happens. But you can see those green bars are already up to 30% severity in 2018 and less so, but also present in 2017 and up to Alaska. So, in kind of answering your question, Chuck, our interpretation of this is that part of it is that those deeper populations are less stressed. They're definitely not seeing the temperature stress that the intertidal populations are seeing, and disease is responding to that. We don't know whether there's also the possibility those plants are resistant for other reasons. But it turned out to be a really, really important study because of what's happened to the intertidal populations. To have this early data when those populations were in fairly good shape before they just started to disappear. And so this is kind of our first clue, and I love the title that Olivia chose: Deeper Habitats and Cooler Temperatures Moderate a Climate-Driven Disease. So that's also sort of the clue about where you go to look for refugia or think about resilience in dealing with managing these populations.
One question. So so the San Juans don't look so bad in the intertidal in 2017, and then they get hit really hard 2018. Is that because of the heat dome?
No, the heat dome was 2021. So the blob, this is still sort of washover, I think, from the blob. Yeah. So yeah, Olivia's project is now to look really closely at our full 10 years of data and really try to put this all together with a long time series. Yeah. So it's not just us recording this. We do work closely with the Department of Natural Resources eelgrass team, and they survey over 214 sites. Not as in depth as we do. They don't have like 6 transects per site and hundreds of plants. But they do have pretty comprehensive surveys. They found that the San Juan Islands is a region of concern with 4 times more sites showing declines. Those are the red ones. These are our San Juans, then increases. So we're really being hit with a big regional problem. And heat wave is part of it. We get daytime low tides that we think are kind of amplifying the effect of these heat waves. And so we're again, this is just some more data from the Department of Natural Resources showing these are sites with that are very shallow, less than a meter, 1 to 2 meters, -2 to -3 meters. And you can see that all the losses are in these very shallow ones. These are the sites with declines, are the shallow ones here. And the sites that are showing some increases tend to be the deeper ones. And so again, it's kind of filling in the picture of the importance of these deeper sites. So what I want to talk about is sort of, we'd like to use maybe some combination of hindcasting and forecasting to think about what are the sites that are really likely to be less resilient and to be very, very vulnerable for our eelgrasses, and then also use the health of the plants as an indicator of how well they're doing at those sites. And so that's kind of a future project plan for us. In general, I have an interest in disease outbreaks. I've written this book. If anybody's interested in knowing more about disease outbreaks, it's very easy to get on Amazon. They have them in the bookstore here. I have ones at reduced price in my library. I'm happy to sell to you. But what I did was basically took the perspective that I could use just 4 types of organisms to kind of tell most of the stories, especially related with climate. And so the coral outbreaks are a huge climate driving coral decline, partly through infectious disease, partly through bleaching. And Steve Palumbi will be here next week, and he'll be talking about that. Our sea star outbreak talks about the ecological impacts of losing a dominant species, also as a result of heat wave stress. Abalone, there used to be 8 species of abalone in California, and I think it's now 3 of them are on the endangered species list. And the withering syndrome, which is a rickettsial bacteria, is also temperature sensitive. And then finally, if nobody cares about these invertebrates, salmon have been heavily impacted by quite a few different diseases, some of which are also heat sensitive. And that's all I'm gonna say about that. And I think that's all I have in here. Um, oh, I'll just say one more thing, in case any of you noticed this, because this was a recent study that was picked up by the Seattle Times about our work. They said researchers discover eelgrass superpowers in Puget Sound. And I don't know if you're wondering what those superpowers are, but of course, you know, eelgrass are highly valuable as a habitat, but what we've recently found through outplanting mussels inside and outside seagrass meadows is much lower levels of bacteria in the mussels that are in the seagrass as opposed to outside. And so the superpower that these folks were talking about is the ability to detoxify bacteria. And we don't know all of how that's done, but we think part of it is the oxygenation from these plants. So I just want to conclude by just launching some topics for discussion. And I don't know what everybody's state of mind is, whether it's good to take a little break now and then come back and talk about some of this. Yes, I'm getting nods. So how about take a little break and then come back and we'll talk about these things. Yeah.
Why don't you come back at like 3:05?
Oh, that's a Banfield sweatshirt.
There this year.
Oh, you were?
Some measurements. Yeah, thank you.
Okay.
It's cool, but it's like a collaboration with Julian Science and then University. Korea, BC. These are nice. We learn a lot of from them. Banfield is really beautiful. Yeah, no, we talked about that a little bit earlier, yeah. She's in more different.
But Kurt's well.
Yeah.
No, he's. Yeah.
Curtis.
Oh, oh.
No, it's like on better ways. Yeah, yeah. You build, this year we built a lab there, it's called. Measuring a lot of different. Oh, great. So start. Yeah, yeah, no, that's really good, because I mean, we now have a monitoring station here that's, I don't know, like 10 years of data, we'll probably get 6 years of data here. So temperature, ocean acidification. Yeah. So whale those coming every day. For the coast. Wow, what kind of whale? That was on pop up as well. That's exciting. It's beautiful, and the intertidal is a kind of population. We need to get back there. Last time I was there was 2014. Yeah, right. Yeah, it was. It was, well, I remember so clearly because it was right at the beginning of the big epidemic. And millions and millions of stars and it just started to die. And it changes. It's pretty much same right now. There's the same. Yeah. Great. Thanks. Yeah, so this is just. I guess the benefit of that is that
As opposed to just, we don't have that projector.
Well, it's much better than Zoom. You know, projecting it on the screen. Yeah, that makes it complicated. And this is a computer, right? Because it's got, so it's just like every other, we can set it up like any other computer. So we don't have to type into, projector.
Right.
It's just like any other map. What we have to figure out, I'm sure it's built in, is that it's probably smart enough to know to shut off, to cut out the other people when you're speaking. That's something that's probably built into the system, but we don't know it, so we're ending up. It's not so bad when you're presenting here and everybody has their stuff turned off. When Manu was presenting, we had to have everything turned off. If we had to, she wanted to ask a question, I had to turn him off.
No, I saw that. I saw that.
And so, that's, there's gotta be a way to set the system up to avoid that. We don't have, but, but we seem to kind of know, I mean, it's working pretty well. It's a whole lot easier just being able for you to bring your laptop, anybody can bring their laptop, just one Zoom call, and then.
Yeah. It is sort of strange to lecture and everybody's on the
Well, they don't have to be, because the problem, most of them aren't on these computers.
Oh,
These are just here because the computer.
oh, I
allows-
see.
So they're not watching these screens. If they're watching it on Zoom, they're watching it on their laptop. But they probably are.
Okay. It's funny.
I mean, these are what Bruce and for running this.
These are the new ones, huh?
These are the new ones.
Yeah. Oh, great.
And they set them up really fast. They were able to set them all up in an hour.
I guess I thought that, just like the regular.
No, I mean. You can draw out it, you can draw all sorts of
They're good.
stuff that we have
Oh.
done.
Oh.
And.
So you've already gone around and everybody said a little bit about what they do. It's kind of nice to do that. Like ones that I feel like are waterproof.
Yeah, well,
Oh, okay. Forgot about the remote people. Okay. So, or when do we get the remote people on?
when we stop recording.
Oh.
I'll just turn
Well, I
the.
should have finished that up.
Should finish what
I should have just done this and then finished it. So, I could let the remote people go.
Well, someone will be able to say that. Is your, are you on mute now?
No.
Yeah, they're probably very quick to say.
How many remote people? I know.
So we have 5 on right now. But a lot of people like we are in the middle of the night, you know? Or the ones-
Okay, so plan what to do. I thought I was gonna go through this and then we'll transition. I think, well, I'm trying to.
Watch people thinking in the chat. Or fairly waste necessary to take grass production.
Okay, I'll take that. So, hi, everybody, welcome back. What I think we might do, I want to think a little bit about sort of the relevance of the things you're learning in this workshop for being able to manage for resilience, and also to take any questions. And then I think we'll probably break from the hybrid format if the online people want to leave after that, they may, or they can stay. And also, so in a minute, we can also do questions. So I'd like to just go through this because these are some of the discussion topics. I mean, this is kind of a new job for us. Instead of sort of, you know, monitoring and protecting habitats, now we need to figure out, oh, which are the resilient ones that we need to protect and save, because we can't just plant eelgrass. This is actually a little bit of a controversy or a discussion topic within Washington State about what do we do next. There's a lot of people that want to go back and restore the eelgrass. They want to go back and plant eelgrass where it's died. But the first tenet of restoration is that you need to remove the killing stressor. And we're not going to be able to do that in the near term when it comes to heat waves and when it comes to these kinds of climate impacts. And so I think we need to certainly also at the same time focus very sharply on what is it about good sites or resilient sites. So these are my ideas on that. Obviously the cool big seagrass meadows, again, we're finding some of the bigger meadows tend to be healthier. So if I had to kind of go and pick what were going to be the resilient sites, I would look for deeper sites that were larger, and then I would look at their health. We're finding the same thing for the sunflower stars. There are refugia in the very deep fjords in British Columbia where there's hundreds and hundreds of these endangered species that are doing just fine. And we don't know all the conditions that are accounting for that. But again, it's actually quite successful to figure out where the resilient sites are. And then some degree of sort of hindcasting and forecasting, I think, is going to be useful in this analysis. We think that disease could be a really useful tool for some of this mapping work. One is it can help us figure out where good sites are before all of the impacts to the populations occur. It can allow us to detect refugia, and it can allow us to possibly preserve some of the health services we find in these healthy meadows. And then it's sort of surprising to me that we seriously are considering assisted evolution. The coral people, Steve Palumbi will talk about this in terms of his Coral Futures projects, where they're actively assessing sort of the ability of corals to withstand bleaching events and then selectively breed and propagate those corals. So we're basically talking about the same thing, but in terms of disease resistance and again, heat tolerance. And so it's a whole new ballgame to start thinking about heat resistant seagrasses. And that kind of brings me to a question that Chuck just pointed out to me that was in the chat, which I can't actually see right now, about whether plants need to be cleaned or aseptically cleaned in some way. If you're going to be transplanting seagrass around from place to place, you absolutely need to assess its health before you move it from one place to another, or you're just going to be spreading pathogens around. So you may not need to completely sterilize the plant, because the plants don't really like that. But some, again, identifying the pathogens and then some kind of decontamination protocols are important. We have a decontamination protocol for our eelgrass. I don't know if anybody knows what it is. Actually, this pathogen doesn't like low salinity. So plants in very low salinity places tend to do much, much, much better. Again, some of the Oregon populations, it wasn't just temperature. Part of it was the freshwater input was reducing the pathogen. So it's not a complete decontamination, but it certainly knocks back the levels of the pathogen. That also introduces another problem with heat waves, which are some of the terrestrial effects and drought. And so with declining water in some of our estuaries, plants are becoming sicker because of that. So that's about all I have to say for now. Does anybody have do we have any questions? Maybe first from here and then we'll go to online.
Chat box, you can check with the chat box.
Okay. Yeah. Do you want to read it?
You say that the disease is helpful for the seagrass. So I wondered, is artificial sterilization necessary for seagrass protection?
Yeah, so that was kind of what I was referring to, that if you're going to be doing restoration ecology and moving plants around, some level of decontamination would be relevant. It's going to depend on what it is you're concerned about moving around. You can't really completely sterilize a seagrass. And in fact, you wouldn't want to, right? But because seagrass has a microbiome, you have a microbiome, it's sometimes really bad for you when you take antibiotics. It's not great for seagrasses either to remove their resident microbiome. In fact, one of the projects that we work on is studying the role of the microbiome in fighting the pathogen. And I didn't tell you about that because it wasn't that relevant yet to the heat waves conversation. Yeah.
I seagrass for their own habitat. How are they responding to the seagrass waste?
Not very well. Like, it's really impressive, the levels of fish that are in these seagrass meadows. And as part of our project, we've been doing full eDNA analysis, which gives us a species list of the fish in these meadows. And so we haven't actually looked at how that changes, but we should be able to. And I'm kind of looking at Olivia because she's collected a lot of that data.
Yeah, I'm not sure about our local fish communities just yet. I haven't dove too deeply into the eDNA data, but just for context, historically, there were seagrass waste and disease outbreaks in the 1930s and 40s along the Atlantic coast of the US and throughout Europe. And from that, there were devastating ecological consequences. So crashes in shellfish industries, but also crashes in waterfowl populations that lived and migrated and fed on eelgrass. It even led to the extinction of the eelgrass limpet because there was no more eelgrass at so many of those sites. So we have pretty well documented ecological impacts in Europe and the East Coast historically, not as much for our local meadows, but something we're hoping to get into.
Yeah, so we're actually really excited about the eDNA data because we took it before the eelgrass disappeared and after. And so, I mean, it's pretty obvious that there's not going to be any fish there if there's no eelgrass, but it be nice to quantify which fish have disappeared and which ones were there, but I don't remember. There's probably 30 species of fish at least in an eelgrass meadow, even in a small one. And hundreds and hundreds of individuals. So in addition to all the epifauna, all the invertebrates, all the crabs, herring are actually rely on spawning for eelgrass. So it's a very, and baby salmon grow up in the eelgrass. So